We're very excited that we finally have state funding to support the Cranberry Health Research Center once again. It's been quite some time. I'm here with good news today. So some of you have heard me talk about our colon cancer studies over the past several years. We have a publication in press. This is from the work that we did in collaboration with Hong Shao in the Department of Food Science at UMass Amherst. And the results from the mouse study were very promising. What we were able to demonstrate was that mice eating a reasonable cranberry diet with whole cranberry powder at a level that would be considered reasonable for a human being. What we were able to see in this mouse model was a 50% reduction in colon tumor number, size, markers of inflammation in the tissues, and impact on a number of different mechanisms associated with colon cancer, including metastasis, which is a common occurrence with inflammatory colon cancer. So we have some evidence that cranberry has these benefits to the colon, but there's also emerging evidence that cranberry interacts with the gut bacteria. And so a lot of research in the nutrition and health field now is looking at interaction of food components with the gut microbiome because it impacts so many different disease states. So probably 2 years ago, we formed a collaborative at UMass Dartmouth, which we call the Colon Health Initiative. And this is myself working with two new colleagues from the Department of Bioengineering, Vani Bucci and Milana Vasudev. So we're kind of a multidisciplinary team. And what we're trying to do now is focus a little bit more on not just colon cancer, but all the interactions that go on between cranberry and your gut bacteria and how that affects various disease processes that could occur in the body as a result of consuming cranberry. So what we were able to do with the fiscal year 18 funding, we had a very short window with which to spend the funds. It came in March, and we were required to spend it by June 30th, so we weren't allowed to extend beyond that. But what we were able to do is buy equipment, yay! We were also able to fund some students, at least starting on these projects at the beginning of the summer, buy supplies for the laboratories. Just to tell you a little bit about the equipment and what we're hoping to do with that, we got a sequencer, which is housed in Vani's lab. So Vani is a computational microbiologist. He's studying changes in gut bacteria. He does some bacterial engineering. And what they were able to do this summer was to harvest some fecal bacteria from some donors and begin sequencing that bacteria to get a picture of what a healthy human gut looks like. And our next step is going to be to look at how cranberry compounds interact with that bacterial community and what are the associated changes that we can see. Beyond that, what we're hoping to be able to do is to target these compounds to colon tissues, colon tumors as they arise. We're looking to better understand the interactions between cranberry compounds and bacteria themselves. Because some of the cranberry compounds are not that bioavailable, so they get eliminated by the body pretty quickly, we're also looking to develop methods to target those compounds and extend their lifetime. So Milana Vasudev, who's also a bioengineer at UMass Dartmouth, she works on designing nanosphere encapsulation for therapeutic molecules, which basically is hopefully going to target those compounds to the tissues that we're concerned about. So we've begun looking at how to encapsulate cranberry compounds of interest that we isolate in my laboratory. So going forward, what we're hoping to do is in 2019, Vani will bring a mouse model to campus, hopefully, and we'll be able to start looking at some of these effects in vivo, which is very important to do. The initial studies that we're able to do at this point are mainly in the cell and bacterial culture, but we want to begin looking at animals here at UMass Dartmouth. And go from there. In terms of getting preliminary data for an application to NIH, we really do need some animal data. So we're looking forward to 2019 to focus on that.